Dear students, good evening to both of you. I'm very happy to take this class or have discussion with you. It is 10th of February 2025 and time is about 5:40 p.m. So welcome again to this class. Today we are going to discuss factors affecting plant growth. We want to know what are the reasons, what are the causes, what are the factors that one farmer has good growth of the crop and other farmer may have poor growth of the crop. So we would see the reasons or the factors that affect the plant growth. So plant growth is very, very important because you can see first of all, let us see the learning objectives of this lecture. You will be able to understand the meaning of plant growth, what is plant growth, and what are the factors affecting it, and also understand the influence of different factors that will affect the growth and yield of the crops. This is very, very important topic, and some terminology I would like to share. For example, base temperature. Base temperature is the lowest temperature threshold below which development ceases. Means plant will not do anything. It will not die, but it will not do photosynthesis. It will not do biosynthesis. It will remain still. Means the lowest temperature or lowest limit of temperature where the development process ceases is base temperature. Normally for tropical plants, it is considered as about 10 degrees centigrade temperature. If temperature is lower than 10 degrees centigrade, then they will not grow. The growth will not happen in tropical plants. Biotic factors. Biotic factors means factors related to living things. An aspect of environment related to organism or their interactions. Now, diversity. I talked a lot in the last class. What do we mean by diversity? Diversity means the number of species or variety of species present in a field, present in a region, present in a pond, present in a state, or present in a country. It depends. So number or variety of species in a location, community, ecosystem, or agro-ecosystem, or degree of heterogeneity. Degree of heterogeneity of biotic components, means living component of an ecosystem or agro-ecosystem. How many different varieties or different species of living things, including plants, animals, and your microbes, all variety of lives. Now, genotype into environment interaction. This is also important. Many times, genes are there in the plant, but they are not expressed. They are not expressed because environment may not be favorable. Sometimes environment may not be favorable. So environment also influences the expression of the genes. Everybody needs a proper environment, good environment for good growth. For example, you have hybrid varieties. Hybrids are there. Hybrid rice is there. And you know, hybrid rice has very fast growth. Number of tillers are more. Yield is also high than normal varieties or high-yielding varieties. In comparison to high-yielding varieties of rice or any other crop, the hybrids of respective variety have more yield potential. But if your soil is not having enough supply of nutrients, enough supply of nitrogen, or enough supply of water, then you cannot realize higher yields even from the hybrid. That means environment is very, very important. You should get all the resources that are needed by the plant in the environment. All the resources should be present in optimum amounts, or these resources become factor. As I was telling you yesterday, that factors, resources, inputs, they have similar meaning. Only we use them differently at different places. Otherwise, meaning is same. For example, I repeat, soil is a natural resource. Okay, natural resource, but soil is also input. Soil is also input. And then we can see how it is a factor. It is a factor of production because there are 4 factors of production: land, labor, capital, and management. So land includes soil, land includes light, land includes air. My dear students, in case of economics, if somebody asks you what are the factors of production, then Palash, you need to reply there are 4 factors: land, labor, capital, and management. And Palash told me it can be entrepreneurship or it can be risk also. So, or organization also, you can call it by different names. So land does not mean just soil only. Land include other natural resources also, which may be soil, which may be water, which may be light, which may be air also. This oxygen comes under land. In economics, the natural resources are part of the land. So therefore, this environment also affect the growth genotype, and also environment can also affect the development. For example, there are many cases in fishes. In fish, if the young tadpole or if the young fish stays with mother, she will convert to a female. If that it is in certain species of fish. If that fish or tadpole stays with father for a longer time, then it gets converted to the male. So such thing also happen in nature. Environment can affect the genetics also. So genotypes and environment interaction is always there, where environment influences the genetic performance. Performance may be with respect to growth and yield. Now, phenology. Phenology is a study of sequence of development stages of a plant and how it relates to climate. That is known as phenology. Means phenological stages are actually suggesting some development stages or kind of development. Let me teach you here or tell you here what is difference in growth and development. Growth means it is permanent and irreversible change or permanent and irreversible increase in dry matter or size of an individual is growth. You will get, it is written in the next slide. After a few slides, you will get the definition of the growth. And development is phagic changes. Change from one phase to another phase is your development. In the beginning, suppose I am a human being, suppose. So I know how my body was made. A sperm came from my father, which was a single cell. Another ovum came from my mother, which was an egg, which was also a single cell. Then 2 cells united and they made zygote. Okay. So this zygote is different. It is different from sperm and ovum. It is different in morphology, zygote, because it has also become single celled. Now this zygote will continue to divide. It will start division. It will divide and make a mass of cells. That mass of cells will be embryo, isn't it? That mass of cells will be embryo. So from sperm, I have become zygote, and then from zygote, I have become embryo. And from embryo, I will get differentiation. I will get differentiation. This mass of cells will be converted into legs, into face, into my stomach, into legs. It will make different body parts of an individual. And then again, I will grow. The growth will occur, and then I will become a baby. I'll take birth, I'll become a baby. So these are my phagic changes. Pyara Singh, एक गिलास बड़े गिलास में गुनगुना पानी दे गरम और ताला मैं बंद कर दूंगा. So you see, and then from baby I'll become an adolescent, Kishore Avastha, I will become adolescent. And when I will start growing mustache. मेरी growth हो रही है. I'm increasing in height. My growth is happening, but my phases are also changing. My phases are also changing due to growth. After this adolescent, I will become adult. And then after adult, I will become old. And then again, I will die. And my body will be decomposed again and so on. So you see, these are development phases. This is known as phenology. Studying the development phases or development stages is your phenology. So you see, these are phenological changes. These are development related changes, which is called as development. So from sperm, zygote, embryo, and then differentiation, I became baby, then adolescent, then adult, then old, then died. So these are all phagic changes in the life of an individual. Exactly same happens with the plant. But we give different names. We say it is germination, then it is seedling. From seedling, it can go for tilling. There is increase in height, increase in diameter, increase in weight, or increase in number of leaves. But the stages may be different. Then it will start heading, flowering, fruiting, grain development. So this development is achieved by growth. Development is achieved by growth. So growth and development result hand in hand. So because of growth, I am getting development. So I hope that now you get very much clarity about growth and development. Growth is just increase in dry matter of an individual. Simply increase in dry matter of an individual is your growth. Or if one is increasing in size, then also it is growth. Or if one is increasing in number, then it is also growth. So now you see another term is photoperiod sensitivity. Requirement for a minimum or maximum day length for reproductive phase induction. We got plants classified based upon the day length needed for flowering. Day length needed for flowering. We have short day plant, and day neutral plant. Short day plants flower only when they get less than the critical period of day length. And these long day plant flower when they get more than day length, more than the critical period. So that critical period will vary from species to species. So most of the plants are sensitive to photoperiod for flowering. Now, phyllochron, it is rate of appearance of leaves on a shoot. What is the rate of production of leaves on a shoot or on a plant is called as phyllochron. You need not to write what is written on the slide because you will get all the slides. Don't worry, I'll share all the slides. Now, there is one more term which is plant vegetative and reproductive plasticity. This plasticity means ability of an individual plant within a crop. Ability of an individual plant within a crop to modify the number of vegetative and or reproductive structure depending on the amount of available resources. So this is plasticity. Plasticity means the flexibility. Suppose now a plant is getting all the resources required for the growth. All resources are available for the growth of the plant. But suddenly some resources are in less supply. Then certain plants have capability to mature or to complete their life cycle even under the shortage of certain resources. So this kind of characteristic of certain plants is called as plasticity. It may be with respect to vegetative growth, it may be with respect to reproductive growth. So I read it again for your understanding. Ability of an individual plant within a crop to modify the number of vegetative and or reproductive structures depending on the amount of available resources. You know the classical example of pulp millet. Pulp millet is very classical example. If there is shortage of water, so the term is avoidance, escape, and what is that? Tolerance, I think, something like tolerance. So it escapes the escape. Yeah, it escapes the drought. Maybe because of shortage of water, so it shortens its life cycle, and still it will produce ear, and your grains may not be high yielding, but something it will produce. That kind of characteristic is plasticity. Like phylochrone, you have profile. A plant structure resembling a leaf as a bracteole. You know, bracts are smaller leaves or stipules on the base of some leaves. Similarly, you have profile. It is just like bracteole, shorter than bract, or consisting of a modified or rudimentary leaf. Very smaller size bracts. Now we come to the growth. How to define the growth? There are a number of definitions of growth, but what definition I put here is one of the best definition for students and easy to understand. See, number one, you should remember this definition word by word. Growth is a permanent and irreversible change in size and volume of a living structure with an accompanied increase in dry weight. So means dry weight is increasing. If the size and volume of an individual is increasing, but at simultaneous increase of dry matter, accompanied by dry weight. Dry weight or dry matter is same thing. It is one definition, perfect definition. Number two, it describes irreversible changes with time, which are mainly in size. So increase can be in respect to size. However, this may be measured. Like plant height, it can be measured. The length of the branch, it can be measured. It can be also in form and occasionally in number. Number, for example, if you have 2 tillers and suddenly you got 4 tillers. So you can say the growth is happening because from 2 tillers, they have become 4 tillers, 5 tillers. So tillers are also used as a criterion to measure the growth. We'll see how growth is measured. Any doubt in this?
No, sir.
Now, you see, just I repeat what I have shown you before. Yes.
Sir, in previous slide, there is written the increase in form. What does it mean, increase in form?
Here, increase often in form. Form means, suppose it is vegetative phase. It is vegetative phase, it is having leaves only. And then suddenly you get flowering structure. You get spike, you get cob, you get something else. Then it is changing the form. Is it okay? Change of form from vegetative phase. It can have spike, it can have panicle. Then you can say there is change in the form.
Okay, sir.
Is it fine? Often in form.
Yes,
they
sir.
change from form. Change in form.
Hmm.
Now, this is already explained to you that you get zygote, fertilized egg. Then this zygote will develop into embryo. I gave you case of animal, human beings, my own case. But here it is case of the plant. So, you know, fertilized egg, this zygote will result in a fruit. You know, it will result in a fruit or it can result in a seed also. And then the seed will contain your young baby inside it. You know, it can have embryo. The seed will have embryo. And then this embryo will germinate. It will give you germination. And then from germination, after germination, it is seedling. Now it is changing the forms here also. You see, here it was simply egg. From egg, it has become seed. From seed, after seed, it has germination. So it is change of the form also. Here also, we are getting change in form. So from seed, it has become germinated seed. From germinated seed, it has become a seedling. So, and then after vegetative growth, the seedling can convert into an adult plant or complete tree or complete plant. And this plant can convert into flowering and fruiting in plant. And then again, it will give you seed. So you can see these are phagic changes. These are phagic changes in the life of an individual. And these changes are happening due to growth. Because from seed germination, seed has taken water and it has germinated. And in the beginning, it will eat food from the seed. And then seedling will start getting food from the soil. And then it will start photosynthesis, it will grow. Initially, seedling will have 2 leaves. It can have then 4 leaves, 5 leaves. So again, even with seedling is also changing form. From two leaves, it can have 4, 8, 10. And if it is tilling crop, then tiller 1, tiller 2, tiller 3, 4. So you can see these are phagic changes, or these are changes in the forms of an individual. And it is happening because of growth. So growth triggers the development. अगर ग्रोथ ना होती तो डेवलपमेंट नहीं होता, क्योंकि सेल्स अगर नहीं बनती, नई सेल्स नहीं आएंगी, ड्राई मैटर नहीं पड़ेगा तो ग्रोथ नहीं होगी, तो डेवलपमेंट नहीं होगा, फिर तो सीड ही रह जाएगा. Now, and how this growth is happening? How growth is happening? How plant is increasing dry matter? It is the growth of an organ or individual organism occur in 3 stages. So you see, as I was talking to you about zygote. Zygote is 1 cell, and then it will have mitosis. And through mitosis, it will make 4 cells, 8 cells, 16 cells, 32, and go on multiplying. I think you know, 2 power n, pata nahi kya formula hota hai, I forgot. So it will multiply. And so it is cell division. So because of cell division, the growth is happening. Then cell enlargement. Cell increases in size also. So because of increase in cell size, growth happens. And then again, cell differentiation. Sometimes there may be mass of cells. And this mass of cell can develop into a tiller. It can develop into a root. It can develop into any other part of the plant. As I was telling you that from embryo, differentiation happens and different body parts are made. Similar cell differentiation, through cell differentiation, you can have different kind of cells and different kind of tissues. You see, within the roots, if you remember, the root structure. In root structure, if you have horizontal cut of the root, so you see outside you got root hair, then epidermis, and then you will get cortex, then you will get hypodermis, endodermis, then pericycle, pata nahi kya kya, I'm not correct, but I'm just telling you that root is one. But because of differentiation, some roots are doing, some cells are similar kind, making cortex, some are making pericycle, some are making xylem, some are making phloem. So it is just differentiation of cells. So cell differentiation, structure of the cell changes to perform specific functions. Structure is changed. Originally, all have originated from 1 cell. You know, एक egg था, एक जाइगोट था, जाइगोट से ही सारा सेल्स बना है, और लेकिन अलग अलग तरह का हो गया. Some cell look like cortex, some look like hypodermis. Cell ही तो है सारी, xylem is also cell, but specific requirement was there. Because of specific requirement, they have changed their structures. Cells have changed their structure, and it happened mainly because of hormonal roles. Some hormones play a role in differentiation. How come 1 kind of cells have become 10 kind of cells? So it is influence of hormones, perhaps influence of hormones in plants. So you can see cell differentiation, structure of the cells changes to perform specific functions. The similar type of cells having same functions form a group of tissues. So within roots, you have variety of tissues or variety of cell. Similarly, within leaf, within stem, you have variety of cells. So cell differentiation is also causing growth. So there are three reasons for growth. Number one, cell division. Number two, cell enlargement. Number three, cell differentiation. All these three are causing development and growth and development. I hope that by now you understand very clearly difference in growth and development. Development happens mainly because of growth and differentiation. When growth and differentiation happen together, then development happens. Okay? Now, you see bacteria and algae. In bacteria and algae, entire body grows. Mostly these bacteria are single celled. So in this case, entire body grows. In higher plants, growth occurs in cell of growing region. Cell of growing region, you know, apical tissues, apical meristem. So growing region, which is called as shoot apex or root tip. These are the two growing region of the plant. Because of this, plant increase in length or height, and roots increase in length from the tip, not from the middle part or any other part. So growth is always from the tip of shoot or tip of root. Lateral growth, side growth is by the side growth, the stem or the plant increases in thickness, and from the apical growth, it increases in length. So you see, in higher plant, growth occurs in cell of growing regions, shoot apex or root tip, and close to the lateral sides of the stem or root. So, for example, one question is asked, today your tree is 5 meter tall, and it is increasing in height 1 meter every year. 1 meter every year. And you have put a nail, you have inserted a nail horizontally at 3 meter height. So after 10 years, what will be the height of the nail on the stem? Palash, एक पेड़ है 5 साल का, it is 5 years old tree and 3 meter height, soil से 3 meter ऊपर, you have put a nail, एक कील गाड़ दिया उसमें, okay, and its height is 5 meter, और कील आपने 3 meter पे गाड़ा, और 1 साल में 1 meter growth होता है. So what will be the height of the nail after 5 years or 10 years?
3 meter.
3 meter. It will not change. क्योंकि ये length जो बढ़ती है ऊपर से बढ़ती है, apical से बढ़ती है. Whatever length or height has been achieved will remain same, but the growth will occur from the top of the plant, from the top of the shoot. Similarly, in the root, from the tip of the root. But how it is getting in thickness? Lateral growth. एक तो लंबाई में होती है, एक side growth भी होती है. Stem initially is very thin in size, but stem can become very thicker. Tree का stem देखा है. So lateral growth is also there in some plants, and because of that, growth occurs. So you see, growth is in two directions. One is upward or lowerward in roots, or sideways. So now there is one more kind of classification of growth. See, growth can be classified as determinate growth habit and indeterminate growth habit. You should know what is the difference between determinate and indeterminate growth habit. So determinate growth habit is initially plant will grow to a certain size, certain height, and then stop growing due to presence of terminal inflorescence. So plant at first will complete its vegetative stage, and then reproductive stage will start, and it will mature simultaneously. So such kind of growth is known as determinate growth habit. And indeterminate growth habit, in this case, the main stem or main plant will continue to grow, it elongate indefinitely without being limited by a terminal inflorescence. So here the terminal inflorescence will not come, Here the branching will happen. This branching and production of leaf, flower will overlap. So initially vegetative growth will be there. Then after certain period of time, the flowering will start, and simultaneously vegetative growth will continue. So there will be overlap of vegetative and reproductive growth. So this happens in cowpea, for example. In some pulses, you can see the indeterminate growth habit happens in pulses like lobia or cowpea. You have indeterminate growth habit. So here flowering and vegetative growth can go hand in hand. They can happen together. Such kind of growth is called as indeterminate growth habit, which is mostly found in certain leguminous plant. But our most cereal plants like rice, wheat, barley, they have determinate growth habit. Now, you see, there are growth curves also. Let us understand what is growth curve. You have seen that plant grows slowly at the initial stage, and then growth is fast, and then slows down. The rate of growth varies with the stage of the life of an individual, of a plant. So now you see growth rate of plant varies with time in life cycle. Initially slow, then can be fast, and again slow down. Plotting the increase of cell number with time. A typical cell, if cell number is the measure of growth, then you can plot it against the growth or dry matter. So it gives you sigmoid growth curve. There are three phases: lag phase. Initial phase, rate of growth is very slow, or it can be negative also. It can be negative also, or lower growth rate, mainly because of consumption of food material by the germinating plant. It can eat and dry matter, and it can respiration. So there may be a negative kind of growth. Lag phase. Log phase is rapid growth and highest in entire life cycle. So it is. So you can see that there are three phases: lag phase, log phase. Log phase is very rapid phase, and then comes the stationary phase. The rate of growth starts decreasing and finally stops. So this is how you can see the sigmoid curve. So here you can see initially there is lag phase. The rate of increase is very slow, or it can be negative also here. You see seed germinating and losing the weight. So initially means whatever total weight was there in the seed, that may decrease for the time being till the seedling is going to receive food from the soil. Then this is your lag phase. Then you will have slow growth, and then log phase. At this phase, from this to this, it is very rapid phase. And then it becomes stationary, or it can fall. This is sigmoid growth curve, which you understand, I hope. And I will be teaching you law of diminishing return after completing factors affecting plant growth. So it is almost similar to the law of diminishing return, this growth pattern. Now, how to measure the growth? Are there any criteria? Are there any answers to measure the growth? Yes, growth in plants is measured. Yes, Palash. Yes, you are free to ask. Any question, Palash?
No, sir.
Okay. Now you are seeing this slide, measurement of plant growth.
Yes, sir.
Okay. So how to measure the growth? I can measure the growth in dry matter accumulation in plant parts. How dry matter accumulation is behaving? Is it increasing or is it decreasing? If organic matter content is increasing, that means our plant is growing. And you know, students, you will be doing your thesis also. In MSc, I think you will have some thesis work also. Will you have thesis work?
Yes, sir. But it will
Yes,
be
sir.
started from semester.
Okay. So whatever work you will have in agronomy, you need to record dry matter at every, say, 30 days. If your crop is rice or wheat, you have to record data on dry matter production, say every month or every 30 days, every 40 days, you will be recording the data. You will be also recording the data on leaf area index. Normally, we record data on leaf area and then we calculate leaf area index. You will be recording data on plant height regularly, how the plant height is changing. So all these measurements are actually measuring the growth, that over time, after 1 month of sowing, dry matter was so much. Then after 60 days of sowing, how much change is there in the dry matter? And then after 90 days, how much is the change in dry matter? So we observe, we make observations, we record these kind of data to record the growth. We record LAI, dry matter, and there are certain indices which are also used to measure the growth pattern. For example, you can use relative growth rate. So you need not to bother right now, but you should know formula to calculate relative growth rate. Similarly, net assimilation rate, or there may be crop growth rate, CGR. So there are 3, 4, 5 indices that are actually measuring the growth. Many times students do not know what for we are recording these data. These data we are recording that what was the pattern of the growth during first 10 days, first 30 days, then. So over time, how my plant has grown, what was the pattern of the growth? So that is the purpose. So, dry matter accumulation, just from dry matter accumulation, I can work out RGR, CGR, NAR, etc., etc. Increase in height or length or breadth. Sometimes we measure height or length of the plant or length of the branches, etc., etc. Sometimes we measure the length of the leaf also, or breadth of the leaf, number of leaf production also. Leaf production means in 1 week, how many leaves have been added to maize plant. In 1 month, how many leaves have been added to maize plant. In 60 days, and so on. So increasing number of leaves will also tell us the story that it may be growing, plant may be growing. Increase in volume. So sometimes we can measure volume of certain plant parts. Like in case of root, when you make root studies, so how do we measure root? So root growth is measured by root volume, very specific to root. So it is very easy to measure root volume. You can get one cylinder, measuring cylinder. You can put some amount of water. Suppose you got 1 liter cylinder, measuring cylinder, and it is full of water up to 1 liter mark. And then you can put the root into the cylinder carefully. And then some water will come out, some water will overflow. And you can collect that water in a pot and then then measure the volume of the water. So whatever volume is there for that water, this is volume of the root. We know fundamental law of physics. पता है ना volume का, जितना उसके अंदर डुबोगे, पानी बाहर आएगा, that is the volume of that particular matter. So it is very easy to measure root volume. But now scanners are available, machines are available to measure root volume. So root volume may increase or decrease with time. The change in root volume may increase at increasing rate, then decrease. So we can record the data on root volume with time. With advancing time, we can record the data. So increase in volume can tell us whether plants are growing or not. But there are many more measures or parameters for measuring the root growth. You can measure the root length. Total root length can be measured. The root diameter can be measured. The dry weight of the root can also be measured at different stages. So we can record number of data, number of observation on root growth. For dry matter, other aspects, we are recording data for shoot growth. But we need to record sometimes data on root growth also. Increase in leaf number and size. Sometimes leaf size can increase. We can measure the leaf area to see how much is the leaf size. And increase in branching and tillers. We can know that how many branches have increased For example, in chickpea, chana dekha aapne, Kajal beta, chana, have you seen
Yes, sir. I have.
Matar, yeah. Dekha hoga, mustard dekha hoga. So these are branching crops. So normally, initially there may be 1 or 2 branches, but with time, branches can be 4. Primary branches can have secondary, tertiary, and so on. So then the increase in number of branches will be there. So therefore, in certain crops, if we want to measure the growth, we can measure the number of branches also. So this is also a parameter to measure the growth. Similarly, in rice or wheat, we can measure the number of tillers per unit area. Today, there may be 10 plants. Tomorrow, there can be 30 plants. Day after tomorrow, there can be 400 plants in 1 meter square area. So I can count the plants in 1 meter row length. I can convert them in meter square. And then I can convert it into hectares. This is how we do sampling. So increase in branching and increase in number of tillers also can be used to measure the growth. So it is not just dry matter which is used to measure the growth. We can use certain plant parameters also to assess, to measure the growth over time. We need to measure it over time so that we can get a sigmoid curve. From starting from, say, germination or field emergence up to harvesting, we can measure the growth, or we can measure different growth parameters as far as feasible. So this you are going to do in your MSc dissertation or MSc thesis. Then you should not ask your chairman or chairperson, sir, why I am measuring dry matter, why I am counting the leaves, why I am measuring the leaf area. It is just to see the trend in growth over time. And then we can relate this growth to inputs also. In some plots, you will add fertilizer. In some plots, you will not add fertilizer. In some, you will apply water. In others, you will not apply water. So you will maintain different fertility or different water content in different plots. Then you can compare the growth rate. Under which treatment, under which practice, the growth was faster. Under which practice, the growth was slower. Under which practice, the growth was highest. And under which practice or under which treatment, the growth was least. So this growth has very important values in our research work also. Now, there are many more measures to measure the growth. Like fresh weight can be measured. You can take fresh sample and just take the weight, and then you can dry it in the oven. You can dry it at 70 to 80 degrees centigrade for 21 to 48 hours. You can know the dry matter. And sometimes you can put, if it is soil sample, then it is normally 105 degrees centigrade temperature. But for plants, normally we keep about 70 to 80 degrees centigrade temperature, and it is dried for 21 to 48 hours. And then it will not lose more weight. That particular point is your last point when you can take out samples from the oven. So your drying is complete. Then we can measure the length of different organs. As I was telling you, that shoot length or height can be measured. The branch length can be measured. The root length can also be measured. The leaf length can also be measured. Area can also be measured, like leaf area. Leaf area can also be measured. And also root surface area. We can also measure the root surface area. There are a number of parameters that can be studied on the roots. Root length, root dry weight, root volume, diameter of the root hair, or sorry, average diameter of the root, and some calculated parameters can also be measured, like different ratios can be worked out. Ratio of root length to root width and other parameter, we can measure the ratios. Here I wish to tell you one point, that now machines are available to measure the root growth. Some rhizotrons are available. These are the equipments which are used to measure the root in situ, means within the field. You can measure the root growth by using rhizotron equipment. And now currently some scanners are available. normal scanners at which you scan your images. So some root studies can be made on root scanners. So they can automatically count your root length, they can count the number of roots, root diameter, variety of parameters can be recorded from the root scanners. So any student interested to work on roots, you can demand one instrument that is known as root scanner. So you can make a demand to your professor, to your teacher, sir, we want to do some root studies, provide us one root scanner. So because your center or institute is getting newly established, so you must be getting some money to buy new equipment. So you can make a suggestion, we want root scanner. It costs around 11 to 12 lakhs rupees, but it is a good instrument to work on. Now you see, overall, you have seen what is growth, what are different growth parameters, how it can be measured. I tried to explain you all these. Now question comes, why we are studying the root growth, the growth of the plant? What is the purpose of studying the growth? So actually, we want to optimize the growth. We do not want too little growth. We want optimum growth. We do not want very less, very small growth. We do not want very high growth, maximize growth. We don't want maximum growth. We want optimum growth. And optimum growth is the growth where you get maximum output of the economic product, where you get maximum output or yield of the economic product. That growth is desirable. We want to find out that level of dry matter production by the plants which give us maximum economic yield or maximum grain or maximum tuber, maximum fodder with good quality. Sometimes quality also comes into picture. If you go on increasing the growth, then quality, we may lose quality. For example, if you want more yield of wheat plant, there are certain wheat which can give you more and more yield, but protein content can decline in the grain. You can increase the growth of some other plants also where quality can be compromised. So the main purpose of measuring growth is to optimize the growth. And this growth, because we want the most economic yield. Why we want most economic yield? Because we want more profit. We want more returns. Why we are growing the crops? What for we are growing the crops? We are growing the crops to get money. Farmer is growing the crops to get money, to get profit, to get income. So we need to optimize the growth at a point where farmer will get maximum economic return. So that is the purpose of studying the growth. And if the growth is too small, it cannot yield higher. And in otherwise also, if growth is too high, then the cost of production will also increase. You can increase the growth by applying fertilizer, more water, more nutrient. Up to some extent, you can increase the growth. But that does not mean that you will get more economic yield. Take the case of chickpea. I can apply nitrogen from outside also. Although chickpea is a leguminous plant, it fixes nitrogen in the soil. I can also increase its growth faster if I apply urea from outside. You can do it. You can apply urea from outside. The growth will be more. In case of chickpea, you do not go for nipping. In chickpea, nipping is recommended. You can remove some leaves, some part of the chickpea plant so that branching is increased. So in case of chickpea, if you get excessive vegetative growth because of not doing nipping or because of nitrogenous fertilizer, then your fruit production will be less. The fruit production, the pod production will be less because vegetative growth has exceeded the limit. So here also, we do not want maximum growth. We do not want too much of growth. Here, we want optimum growth. So therefore, optimum growth is desirable. Okay, so now we understand how important growth is. Can we modify it? Can we make the growth? Can we manipulate it? Can we optimize the growth? How can we optimize the growth? Because we need to look into the inputs also. we need to look into the cost of the inputs also. So we need to find out the optimum level of inputs also. If we can find out the optimum level of inputs, then they will give us the optimum growth. If I know how much water management, or how much water or how many irrigation are sufficient to give better growth, I will use only those irrigation. If I know that this much of nitrogen will give us optimum growth, I will use only that quantity of nitrogen. If I know that 2 liters of chlorpyrifos is enough to control this insect, I will use only 2 liters. I will not use extra. So here, economic concerns come into picture. Economic concerns. So we need to know the resources or factors or optimum level of factors so that we can get the optimum growth. So here, therefore, we need to study the factor that will affect the growth, because we can optimize the level of the factors to achieve the optimum growth. So therefore, we should know what are the factors, what are the important factors that are going to affect growth of my crop, growth of my plant. So therefore, we need to study factors affecting plant growth. As already told you, that genetics and environment, they influence overall growth. They also influence overall yield, because yield is the result of growth. So first we need to achieve the optimum growth, then only we can get the optimum yield. But this optimum growth and yield is also affected by genetics into environment. So genetics is your seed, kind of variety you are using, and environment is your external, or environment is your field. And in field, you have factors. In field, you have resources. I tried to make overall picture why we study the factors affecting crop growth or plant growth. Now you see, dear students, there are a number of factors that affect the plant growth or crop growth. If I say plant growth, you can take it equivalent to crop growth. So farmers can control only 45 factors. And all these factors must operate in unison. You have to combine all these factors. You don't bother about it. I will explain it at a later stage. But right now, you can see there are at least 52 factors, out of which 45 can be managed by the farmers, but farmers do not have any control on at least 7 factors. This is given by Tisdale's book. Now that book is by Havlin. I will send you this book. So factors like temperature, sunlight, storms, flooding, rainfall, carbon dioxide, altitude. Farmers cannot control these factors. They cannot manage these factors. They are kind of climatic factors. Now, just to show you that what the factors that affect crop growth are very large in numbers, difficult to remember, just to give you exposure. Do not bother about it. So you can see the soil properties like organic matter in soil, clay content in soil, CEC of the soil, quantity and intensity of light, slope topography, temperature, irrigation, water percolation rate. There are so many factors that can affect yield and growth of the crop. So it is difficult to discuss each one of these factors, how they are going to affect the growth. So see another case, factor affecting yield and quality of potatoes. This is given by Kunkel et al. So you can see there are a number of factors which can affect the growth. Frost-free period, air temperature, soil temperature, light intensity, day length, fertilizer, seed quality, seed piece size, compaction, soil, variety, and so on. Don't bother about it. It is just to show you that there are large number of factors which affect the growth of the plant. So we cannot study individual factor. So what plant physiologists or soil scientists or agronomists have done, they have grouped these factors. We have classified the factors and hardly 6-7 factors are there. So you can see I made this classification for ease in understanding. So if you should remember these factors. 4 or 5 major factors are there. You need to remember these factors. They are important. Now see from the left side, there are factors. Factors are listed. These are the factors affecting plant growth or factors affecting plant yield or crop yield. So there are broadly 2 factors. One is external factor, other is internal factor. Internal means this is a genetic factor, genetically controlled factor, which is there in the seed, which is about the genes of the seed. So genes of the seed means genetic factors. So there are 2 factors which affect the growth, external factors and internal factor. Internal factors are genetically controlled factor, or you can simply say genetic factors. Now you see in external factors can be further classified into 3. One is environmental factors, means what is happening outside the plant. What is happening outside the plant? What kind of biotic and embiotic interactions are happening in the surroundings of the plant, in the crop field? What is happening? So this is environmental factor, external factor, and then management factor. How farmer is growing the crop? Is he following crop rotation? Is he doing zero tillage? What kind of tillage operation farmer is doing? Is he adding organic manure? Is he controlling weeds? So all kind of cultural practices or agronomic practices, they also affect the growth. Sowing time, depth of sowing, all such factors are management factor or cultural factors or agronomic factors. They are going to affect the growth of the plant. That is why we want optimum spacing, optimum seed rate, optimum time of sowing. All these factors, we are just getting the best of these factors so that we can get the optimum growth. If time of sowing is delayed, you will get slow growth, less growth. If time of sowing is too early, then also you can get wrong growth, maybe excess growth or maybe too less growth. So management factors are your agronomic factors which controls the growth. Number 3 is socio-economic factors, which are at the end of the farmer, end of the grower, whether our farmer is literate, is our farmer skillful, what kind of society he belongs or she belongs, or literacy level. So these socio-economic factors also govern the growth of the crop. If a farmer is having less money, the status, he does not have much money, so he cannot buy fertilizer, he cannot buy pesticide. But if a farmer has money, or if he is having bank facility to borrow money, the growth of his crop will be better than the farmer who is not having money. So money or resources or this credit also affect the growth of the crop. So I hope that external factors are clear to you. Environmental factor, management factor, and then socio-economic factor. Now within environmental factor, there are 2 factors. One is abiotic. Abiotic factor means non-living. And then biotic factor. Biotic factors are your weeds, your insect pest and disease, nematodes, all these biotic factors which can adversely affect the growth of the organism. But some biotic factors can also favor the growth. For example, some useful organisms are there in the soil. Some nitrogen fixation can be there by biotic factors. So biotic factor, we have mixed. Some are deleterious bacteria also. Some are harmful organisms in the soil. Some can cause disease. But some biotic factors may be useful also, which we supply through biofertilizers, mycorrhiza, certain fungi, they are useful. So biotic factors are there, abiotic factors are there. So in abiotic factors, it includes Wants plus light. Wants, W-A-N-T-S plus light. In Wants, W is for water. Availability of water in soil is necessary for optimum growth. So water availability or irrigation is going to affect the growth. Number 2 is air, A for air or atmosphere. atmospheric conditions will also affect the growth, particularly the carbon dioxide condition. Carbon dioxide condition in the atmosphere will affect the growth of the crop. If you increase the CO2 concentration, the growth will be more. If you decrease the CO2 concentration in the environment, the growth will be less because of reduced photosynthesis. So atmosphere is going to affect the growth of the crops. Then N is for nutrients. N is for nutrient supply, means addition of nutrients through fertilizer, biofertilizer, or through organic manures. So nutrients will affect the growth. I need not to overemphasize it because you know how important these nutrients are. Without nutrients, you will get reduced growth. And then T is for temperature. Every plant will need optimum temperature for optimum growth. You might be knowing about cardinal temperature. For every development phase, like germination, tilling, vegetative growth, reproductive growth, 3 different kinds of temperature are required. Lower temperature or minimum temperature, optimum and maximum temperature. These are called as cardinal temperature. So temperature is very, very important for growth. Too much or too less than the optimum can adversely affect the growth of the crops. And then S, finally, wants, easy to remember, you can remember easily, wants plus light. S is for somewhere to grow, means soil. S is for soil. So your soil will definitely affect the growth. If it is sandy soil, on one hand, on another hand, it is clay soil or clay loam soil. So naturally, the growth will be better for clay loam soil than in sandy soil. So soil conditions are going to affect the growth of the crop. So this is how we can classify the factor. And now I will try to explain you all these factors, their effect on plant growth in very brief. And today I was not able to finish this lecture. I will continue this lecture. Last 10 minutes are left. So let me discuss some factors in detail. Now it is genetic or internal factors. I have already discussed this with you. So genes affect the growth. For example, you see eucalyptus tree. Have you seen eucalyptus tree? It has got very small seeds, but it grows very tall. The growth rate is very fast. If you take some grasses or some other plants, in those, the growth rate is very slow. So it depends upon genes, kind of genes that are available in the plant. So all different plant species will have different growth rate. Okay, is it clear? So for example, growth rate of sunflower may be faster than the growth rate of pigeon pea, because this sunflower takes very less time for its growth. Say in 90 days it is complete. But if you take some pigeon pea varieties, they take 270, 300 days. So growth rate, the rate of growth can be slower in pigeon pea than in sunflower, than in eucalyptus. So the rate of growth will vary with the crop species, number one. Number two, the rate of growth will also vary with the varieties, kind of varieties we are using. As I have given you example of hybrids, high-yielding varieties. They have faster growth or more growth than the local varieties in general. These are general points. But we need to remember, if the growth is more, if growth is high, the resource need will also be high. If you get eucalyptus, it is growing fast, it grows more. So it also removes more nutrient. It also removes more water. So similarly, if you have hybrid varieties, the nutrient requirement will be more, the water requirement will be more, the pesticide needs will be more in case of hybrids. Of course, growth is more, growth is fast, but resource use is also more in that case. So we need to supply more resources. The recommended rate of nitrogen, which is there for hybrid rice, may not be same for the variety or same from the local variety. For local variety or like basmati rice, we recommend nitrogen application at 120 kg per hectare. But if it is high-yielding variety, then you need to apply 150 kg nitrogen per hectare. But if it is hybrid of rice, then we recommend 180 kilogram of nitrogen per hectare. So depending upon the variety, to optimize the growth, we need to use graded level of input, different level of input. Similarly, certain variety, long duration varieties will need more irrigation than the shorter duration varieties. So this is how genetics is going to affect the growth. And here we need to manipulate our resources. We need to modify or mold our resources. So the increase in crop yields and other desirable characters are related to genetic makeup of the plant. So this is how we can explain that how genetic factors can affect the growth, and they work in unison. They work in unison means together with the environmental factors. Only genetics, good genetics is not enough. You need good environment also. अगर कीड़ा आ गया, इंसेक्टिसाइड, कोई बड़ा कीड़ा आ गया, उसको पूरे को खा गया. You have planted hybrid maize, and you know about fall armyworm. वो आ गया, वो सारा नाइट्रोजन लगाया हुआ, पानी लगाया हुआ, सब बेकार हो जाएगा. So it can all go in vain. So these factors should work in unison. Means we should take care of all factors. ऐसा नहीं है एक तरफ NPK लगा दिया, फर्टिलाइजर लगा दिया, इंसेक्ट कंट्रोल नहीं किया तो सारा बेकार हो जाएगा. So you can see in genetic and internal factors, you have C3 or C4 plant. So generally speaking, just 4-5 minutes और है काजल, don't get impatient, आराम से बैठो. I know you are tired. All the day you must have attended several. I know, beta, I know. पूरा दिन इतना दिन क्लास किया होगा सुबह शाम तक. You must be tiring now. Just 5 minutes और है. So in genetics, you see C3 plants have slower growth rate. C4 have more growth rate, faster. Now you see sugarcane, you see maize, you see pulse millet, sorghum. They are C4 plant, but your pulses, they are C3. Generally speaking, the photosynthetic efficiency, water use efficiency is higher in C4 plant than in C3 plant. And varietal differences are also there in growth. Yielding ability, early maturity, resistance to lodging. A variety which is not having resistance to lodging, the growth may be there, sufficient or high growth can be there, but it can lodge. Finally, the growth can be reduced, the yield can be reduced, the economic returns can be reduced. So, but this is a genetic character, but it can be influenced by environment also. Excess application of nitrogen can cause lodging. Tolerant to insect pests and diseases. If our varieties or our crops are such which are tolerant because of genetics, if they are tolerant to insect pests and disease, we can get better growth from such varieties without using chemicals. And then quality of grain. As you know, if we want more quality, the yield will be less. Generally speaking, yield will be less. The yield of pulses is low because they contain high nitrogen. The pulse crop may contain 2 to 3% nitrogen. In pulse seed or pulse grain, you can have more than 3% nitrogen. Protein is 20 to 25. So the nitrogen content is high. In rice grain, the nitrogen content is only, say, 1%. 1%. In pulse, it is more than 3%, or maybe 4% in some pulses. So you get 3 times more protein in pulses than in rice. Because rice yield is high, protein content is less, means quality is poor. Pulses have more protein, that means quality is good. So therefore, yield of pulses is low because you want quality. If you want quantity, then the quality is affected. Take the case of potato. Potato has only 0.5% nitrogen, tuber. तो कुछ भी नहीं है प्रोटीन उसमें. बहुत सारा पोटैटो खाना पड़ेगा, अगर प्रोटीन की रिक्वायरमेंट पूरी करनी पड़ेगी. लेकिन जो लोग मीट खाते हैं उसमें 25% प्रोटीन हो सकती है. दादा की मच्छी में तो 30% प्रोटीन होती होगी. पलाश मच्छी में तो बहुत प्रोटीन होती है. तो आप थोड़ी मच्छी खाओगे, हमें चावल ज्यादा खाना पड़ेगा. नहीं तो चावल और मच्छी साथ खा लो तो प्रोटीन बराबर आ जाएगा. So you see external factors are environmental factors. So I'll discuss all these factors tomorrow. I'll continue tomorrow and I'll try to finish these factors, and this is very interesting subject. I'm not just teaching you one aspect of one particular topic, but I'm trying to teach you other aspects also related to one particular subject. Thank you. So if you have questions, suggestions, or any point of view, you are free to speak. कुछ भी पूछना हो, कुछ कहना हो, you are free to speak. We have a few minutes, 1 or 2 minutes left, I think. So any point?
सर, स्कैनर के बारे में थोड़ा बता सकते हैं कि अगर हम सॉइल को डिग नहीं करेंगे तो क्या वो सरफेस ऑफ द सॉइल से स्कैन कर लेगा?
एक्चुअली इसमें हमें कैमरा फिट करना पड़ता है रूट के पास में. राइजोट्रोन में कैमरा एक प्रोब होती है, एक सैम्पलिंग प्रोब होती है, शीशे कांच की बनी होती है, उसको मिट्टी में इंसर्ट करना पड़ता है, उसमें कैमरे फिट कर देते हैं, एंड वो कैमरे लाइव इन सीटू रूट का पिक्चर लेते रहते हैं. पिक्चर लेके लैपटॉप से कनेक्ट कर दिया जाता है, जहां एक सॉफ्टवेयर होता है जो उन पिक्चर का जो फोटो लिया है, जो स्कैन किया है उसने, उसको वो कन्वर्ट कर देता है डिफरेंट रूट पैरामीटर्स. अल्टीमेटली ये भी एक तरह का फोटो लेता है और स्कैनर की मदद से हम रूट का साइज पता कर लेते हैं. और नहीं तो मैं आई विल टीच यू one lecture on root, about root. There I will explain you all these things. So don't worry. I will teaching you about root shoot ratio in detail. In one of the lectures, I will teach you about this. Okay? If our classes continue, I don't know, means when your semester will end, this semester?